So today we will talk about the muscle contraction. In muscle contraction, first we want to look at the anatomy of the muscle. A muscle, this will be our muscle. So in a muscle, then we have these little tube, this mini tube that is connected by connective tissue. Connective tissue, and these tubes are called fascicle. And in fascicle, we have more of the smaller tube. This is called a fiber cell. And the fiber cell is most of our muscle contraction, everything will happen in the fiber cells. And each of the fiber cells, we will have this neuron, motor neuron, that connected to the fiber cells, and then can generate all the force and all the information go from there. So now we're going to zoom in from a fiber cell to see closely what the structure look like. So in the fiber cells, we will have a lot of the T tube. So we have a lot of T tube here. This is called a T-tubular tube. So these tubes are actually the inversion of the cell membrane. So it acts as a rapid transmission for their action potential. Because if we don't have the tube, the action potential will harder to reach and to spread all over the fiber cells. And also together with the fiber cells, we will have some different layer that is called the sarcoplasmic reticulum. So actually these is in other cells we will call it the reticulum, endoplasmic reticulum. So this type tubes originate from the endoplasmic reticulum, but we call it sarcoplasmic reticulum particularly only for the muscle cells. And also in the muscle cell, in the fiber cell here, we also have some little tube right here. And inside the tube, we will have our thick filaments and also our thin filament. And the thick filament is made of our myosin. And the thin filament is made of actin. So really important that our muscle contraction is actually the interaction and the sliding of the myosin and actin. Okay, it's really important that these actions, we have to understand these actions. So how do these work? So from here, we're going to zoom in. So the interaction going to zoom in somewhere from here. It can be interaction. Now I'm going to draw out. So in our fiber cell, this is in our fiber cell, we'll have the T tube. This is the T-tubular tube. Okay, and in D tube, now we'll have, this is the end plate, and the end plate will be the one that touching with our motor neuron. So we can clearly see that I am actually zooming in from this part, this part of the cells. From this part right here, we have a motor neuron that attaching right here and connect to the fiber cells. And in our fiber cells, we have our tube, T tube, and also we have our sarcoplasmic reticulum also. So in here, at the same time, we will have our sarcoplasmic reticulum. This is sarcoplasmic reticulum. So really important thing to know that sarcoplasmic reticulum has a really high concentration of calcium. This is the storage of calcium. So from here now, we will see all the interaction and how it's happened. Also, in our fiber filament, we already know that we have all the thick filament, the thick filament here. This is all of its tail, and then also we have all of our thin filament right here. So now we will see how the action will act on this. The action will act on this is first our action potential will reach our motor neuron, and from here it will release a neurotransmitter. The neurotransmitter right here is actually still our acetylcholine, and when it releases this in our muscle cell, there's one type of special ligand gated channel. This is called nicotinic ligand gated channel. So our actin will bind into this, and from here it will open the gates. This gate will be open. And one special thing about nicotinic ligand gated channel is that it can let both our sodium and potassium can flow in and out from this gate. So as usual, we know that our sodium high concentration outside of the cells, it will try to flow into the fiber cell, and then we have our so I'm drawing this one. Oops, actually nope. So then also we have our sodium will flow out of the cells, and then we have potassium will flow. Sodium will flow into the fiber cell and potassium flow outside the cell. However, really important, we know at this point that the permeability for sodium is larger than the permeability for potassium. That's why a lot of sodium in more than a lot potassium out. So because of this, we will create a depolarization. So when we have a depolarization, this will create action potential, and this action potential will radiate and go to the T-tubular tube. Okay. So when we reach the T-tubular tube, actually in our T-tubular tube now, we have another protein. This protein is really special. It is voltage trigger. It is voltage trigger protein. So it is voltage trigger protein, meaning that this voltage, the action potential, can trigger it and activate it. So when this trigger and activate our protein, this protein really special is connect to this. And by the way, the voltage trigger protein, this is called dehydropyridine gate. I'm not sure, I don't really remember the name, but it is a voltage protein right here. And then we have this ligand. This is the mechanically gated channel. So this is the gate for calcium. This is the gate for calcium. So when we see that our voltage trigger protein is triggered by the action potential, it will act as the force. It pull this mechanically gated channel. So this gate will open. And when this gate open, calcium will flow out into out of the sarcoplasmic reticulum out to here. So when we have our calcium flow to here, now it will bind into our thick filament and will activate and generate the force. So move on from here. Now we need to understand what is happening here that create the force. So zoom in this picture. I know it's a lot of details. Zoom in this picture. We will see that this is our thin filament. This is our thin filament, and it is a G-actin protein. It's made of protein. It had two sides. One side is the binding site, and the white side is our binding site. This is our binding site, and then the other one is not. And also, it usually come together with like two in pair like this. So it's like a two necklace together and show two binding sites. And this binding site is really important that it is where our thick filament will bind into. The thick filament had really strong attraction to this binding site, want to bind to this binding site. And the force it generated by the thick filament, which is our myosin, that walk onto the thin filament, which is the actin. So the blue thing need to bind to the binding side of the red necklace and walk on it to generate the force. If this binding doesn't happen, then there will no force generated. So in general, we will have some the two type protein that will control the force and then control this binding. We'll have the rope right here and the rope right here. This is called tropomyosin. And then we also have a protein, kind of like a yellow protein here. This is troponin. These two proteins are really important in control and regulate the force, and by blocking the binding site. So we can see tropomyosin when there's no calcium, this system will be turned off because we have tropomyosin is now blocking the binding site. So because blocking the binding site, our myosin cannot bind to the binding site, so there's no force generated, no force. However, as we see earlier, when all of this system activate and all of this go around, we'll generate our calcium. We will let the calcium flush out, right? So when we have the calcium now flush out, and we have calcium with the calcium, then this calcium will bind into our troponin. And when it binds to troponin, it will twist and rotate our tropomyosin, to expose and to reveal our binding site. So now our our myosin can now bind into the binding site and generate the force. And that's how the force is generated. So we can see how this system works. Usually we have the binding site is the yellow part that is it will make a cross section, right? This. We'll see that now it is right on top of the binding site, so the myosin cannot bind in. But when we have calcium in and activate the troponin, the system is on, then we will have the troponin will rotate a little bit and expose our binding site. It rotates. So now it rotates to this side or this side. And now we have the exposure of our binding site right here. So now we can have our myosin can act on here and it bind to here and walk along their actin. And generate the force. So in here, in general, we see that action potential would radiate to the motor neuron, release acetylcholine, and acetylcholine will bind into the nicotinic ligand receptor. From that, open the gate for to create a depolarization. Another action potential go to the tube, and this will activate the voltage trigger protein. This will activate and make a force that pull the mechanically gated channel and release all the calcium. And when calcium flush out, it will bind to troponin. Troponin then will activate and rotate our tropomyosin, reveal the binding site, and now our myosin can bind on the actin and generate the force. So, how do this work? How does interaction of myosin and actin? How is it generate the force? So we will have a cycle. This is called a cycle walking of our myosin. So myosin, I will represent the red thing is like our thin filament. And so in general, we will have our tail of, this is without ATP. Like at the ATM, at this point, we will have two things. We'll have this is the ADP, and we have this is at the PI, right? The phosphate group. So when we release the phosphate group, we will create a power stroke. At this point, it will look like this. So now it's going like this. It will wrap along. It will wrap for reaching out, create a power stroke. So with a power stroke, and now we release our phosphate group. Now only we have the ADP. And then when we release our ADP, then we end the power stroke. It's like you moving and you wrap something and kind of like a pedal. So this is the end of power stroke. At the end of power stroke, we already release our ADP. So no more ADP here. And at this point, you will see, really important, no ADP, no PI, no ATP. The actin will really, myosin will bind really strongly to the actin, doesn't release. And that's how dead people had really hard body because of this, does not get release of actin and myosin interaction. And after this, now we want to bind to an ADP, binding of ADP. So when ATP binding, it would release this hard rock binding, right? It release. So now this lifts up, and we have our ATP right here. So ATP right here now bind into. And now from this to this is another cycle. When ADP get hydrolyzed, ATP get hydrolyzed to ADP and PI, then we have it kind of like ready to roll for another cycle. And this go in a cycle non-stop continuously to create the force.